Hello, I'm Susan Tallman. Welcome to a seminar titled Cover Crop Cocktails as Partial Summer Fallow Replacement in No-Till Wheat Fallow Rotations. This is a seminar I originally gave in defense of my master's thesis in April of 2014 at Montana State University. Let's get started. Montana has something that I call the Summer Fallow Challenge. If you're driving across our main grain-producing region, you'll see there's long strips of wheat crop right next to long strips of bare ground. That bare ground is something we call fallow. Fallow began as a means to recharge the soil moisture profile. We discovered that if you grew crops one right after the other, that there wasn't enough soil moisture to do that, and there was decreased yields. So the advantages of fallow is it provides some soil moisture recharge, and there's also a nitrogen benefit. The problem with this soil moisture recharge is that it captures at most 40% of the precipitation that falls during that fallow year, so it's not incredibly efficient. Unfortunately, there's a lot of disadvantages to summer fallow, the big one being loss of organic matter. This loss of organic matter has led to increased soil erosion, decreased soil structure, decreased water holding capacity, the potential for increased saline seeps and increased nitrate leaching, and there's also decreased soil biological activity in that fallow time period. Now we're talking about all of this within the context of the Northern Great Plains, and here is a map of the Northern Great Plains ecoregion, which is about 73 million hectares. It encompasses 2 provinces in Canada and 5 states in the United States. In 1971, summer fallow acreage, the area of summer fallow was at its peak in the Northern Great Plains, 17 million hectares. By 2010, that had decreased to less than 4 million hectares, primarily as a result of the advent of no-till systems, which allow for greater water holding capacity. Within Montana itself, we often look at the Major Land Resource Area 52 as defined by the NRCS. This MLRA 52 contains our greatest wheat producing regions, and it's about 6 million hectares. Of that, 2.7 million hectares are in crop, and every year about 1 million hectares are fallowed. This represents about 25% of all fallow area in the Northern Great Plains, and we feel that this makes the MLRA 52 an excellent target for research in ways that we can decrease fallow. There are 2 basic techniques that can overcome the negative effects of fallow. The first is no-till. By using no-till, we increase the soil aggregation, increase the water holding capacity of the soil, and we decrease the potential for erosion. But no-till alone can't overcome the negative effects of fallow. In order to do that, we also need to grow more crops in the same amount of time, which is known as crop intensification. Now we know that we really can't grow full season crops back to back because of the soil water use, but we can grow some cover crops or partial fallow replacement. The main cover crop that we've been researching here in Montana, Dr. Miller and others have been doing that for over 10 years, has been the use of legume green manures or single species cover crops. These provide a benefit of increasing the organic matter in the soil and giving a nitrogen benefit. There are some challenges in using these cover crops, however, and those are water and time. These cover crops need to be terminated about the time of first bloom so that they don't use too much soil water. In addition, you can't expect to just use these cover crops once and expect a positive result. In fact, if you use them once, you'll probably see only negative results in that they'll use water and soil nitrogen and decrease the yield in the following wheat crop. Research has been showing that you need to have them in the rotation for about 6 to 8 years before you see the positive benefits of the nitrogen and the increased organic matter. So currently, the hot topic that's been out there is what if we use more than one species of cover crop? And a lot of this activity has started with a group of farmers in Bismarck, North Dakota, who are using cover crop cocktails, and this has been catching on. Here's a flyer that the NRCS has recently put out talking about cover crop cocktails, saying that innovative farmers are planting cover crop cocktails of 6 to 12 plant species to get diversity above ground, which then creates diversity below ground and mimicking the microbial properties of the diverse native prairies. And if you look in the popular press, places from Tennessee to Iowa, they're all covering this as a story that cover crop cocktails are a new innovation in agriculture. In 2009, I was working for the ATRA project, otherwise known as the National Sustainable Ag Information Service, as the agronomist, and I got to visit a group of farmers in Bismarck, North Dakota, who are using these cover crop cocktails. They use them for flexible forage and silage replacement, and I was really impressed by what I saw. They're decreasing their herbicide and fertilizer use. They decrease their forage cost, and as a result, they've increased the bottom line. If you're interested in learning more about these farmers, we did create a 1-hour webinar, and there's 4 case studies about some of the farms I visited at the atra.org website. You can also visit the Burleigh County Soil Conservation District website for more information as well. So farmers in Montana have taken notice of the success that the farmers in North Dakota are having, and we're asking the question, can these cover crop cocktails work in Montana? This is an image the NRCS provided, 2011, the different sites around the state that are trying some of these cover crop cocktails, and you'll notice that most of the farmers who are trying them are right there close to the North Dakota border, but not too many up in the MLRA 52. And of course, the big difference when we want to see can something work in our region, we have to look at the differences in precipitation. So I've graphed out here 3 locations: Bismarck, North Dakota, Great Falls, Montana, and Havre, Montana. Both Great Falls and Havre are within the MLRA 52. And across the x-axis on the bottom is each month, and on the y-axis is precipitation in inches. And you can see towards the start of the year that Bismarck and Great Falls have almost similar rainfall patterns, but once we get to the months of July, August, and September, the rainfall in Bismarck definitely outpaces the rainfall in Great Falls and Havre. In our region, at about June 15th, our precipitation shuts off. So in order for these cocktails to work in our region, we need a system that works with our specific precipitation window.
So our study objectives, we had 2 study objectives and kind of a silent third objective. We wanted to compare the agronomic response variables of fallow, pea, and 10 mixed species cover crop cocktails. Excuse me, 9, I think. We wanted to look at their biomass production, their biomass quality, what happened to soil, water, and nitrate after they were grown, and the wheat yield after they were grown. And then we wanted to compare the select soil biological parameters of fallow, 1 pea treatment, and 1 multi-species cocktail treatment. And we looked at 5 response variables: microbial respiration rate, soil enzyme activity, soil temperature, mineralizable nitrogen, and microbial colonization. Kind of our third silent study objective was figure out how to do this research in the first place. Because we didn't have, there's no one we could talk to really to tell us exactly how to do this. We had to do a lot of trial and error that you'll see as we go through some of these years here. So we started out by saying we're going to do our cover crops in the spring window. There was a couple of windows that we were looking at. We decided on spring because historically that's worked best for us with single species cover crops. In North Dakota, a lot of times their window they're using is July through October. But we, with our low precipitation in that window, we decided we'd stick with our peak precipitation window. Once we had decided that we were going to go with the spring, then we decided, well, how do we structure this study? And we started by picking plant functional groups. We picked 4 functional groups: nitrogen fixers, and that would add a nitrogen benefit to the soil; fibrous rooters, which would hopefully add some carbon to the soil; taproots, which might their roots might go deeper into the profile and scavenge nutrients or break up plow layers; and brassicas. We know that there's work being done on brassica cover crops, and they're potentially biofumigation. They can potentially suppress fungi or pathogens that are in the soil, so we thought there might be an interesting angle there. Within each of these plant functional groups, we selected 2 species, and you can see in some years we tried one and it didn't work, and so then the next year we tried something else. But spring pea had to be in there because we've done so much work on spring pea cover crops at MSU. 2012 was common vetch. 2013 we used lentil. We had oat in our mix, Italian ryegrass in 2012, and millet in 2013. Our taproots were safflower and purple top turnip, and our brassicas were daikon radish, camelina in 2012, and winter canola in 2013. So to design our treatments, we designed them around our functional groups. The 3 core treatments of our study were really fallow, pea, and our full 8 species mix that included all 4 functional groups. Then we grew each functional group separately, so those were 4 more treatments. And then our last 4 treatments were taking the full mix and pulling out each functional group. So what was it like if we grew the mix in absence of a functional group? And that constituted our minus nitrogen fixers, minus fibrous root crop, minus taproot, and minus brassica. These all were 6 species mixes, except for the 11th one was a 5 species mix. We left out turnip because although we put turnip in the taproot category, it would have provided brassica functionality because it's also a brassica. I should also say that we got a lot of ideas on this study based from talks with USDA, ARS, and Manda, North Dakota, who's working on similar projects. So how did this work out? Year 1, we had at each site a randomized complete block design with 4 replicates. And you can see all of our 11 treatments are in each block randomized. We planted in the spring, and then we terminated with herbicide in that first year anyway, around June 15th. Then the second year was our wheat response crop year, and we planted wheat across the top of all those, that didn't sound right. We planted wheat. We planted wheat afterwards, and we used 3 fertilizer rates. We had no fertilizer, 44 kilograms, and 88 kilograms of nitrogen per hectare. So this was a split plot design. We had 4 site years, and our site years were staggered. We always had 1 site year in the Gallatin Valley and 1 site year up in the MLRA 52. So starting out, our 2012 cover crop sites were at Amsterdam, and then Conrad up north, and then 2013, our cover crop sites were Bozeman and Dutton. This year, right now, we're going to plant cover crops again at Amsterdam and Conrad, and winter wheat has already been planted at Bozeman and Dutton. I should say, too, that most of these sites, 3 of these sites, Amsterdam, Conrad, and Dutton, are all on private farms, which have been in no-till management for more than 3 years. The Bozeman site, however, is an MSU piece of land that has been tilled. It's a wetter site, and it has very high organic matter. So 2012 seeding, we seeded in early April, and we let it grow till June 15th. Then we sprayed out with herbicide. Our seeding rates were variable. We used, to figure out how much of each species to put into a mix, we said, well, what is its monoculture rate? And then we divided by the number of species in the mix. We separated out large seeds and small seeds and planted those at different depths in alternating rows, 2 and 4 centimeters. And then 2012 happened, which was a record drought. So this is an aerial photo of our Amsterdam site taken at about May 30th, where you can barely see the cover crops coming through. At Amsterdam, our mean biomass across all cover crops was 0.85 megagrams per hectare. And you can see that it really didn't even get up much past the wheat stubble, and there was no canopy closure. This is our minus fibrous root treatment at Conrad. And here we had about 0.4 megagrams per hectare average across the site. Conrad, we had difficulty killing downy brome prior to seeding. We didn't get a good kill, and so downy brome overran all of our treatments that had a fibrous root component. So we were only able to salvage about 5 of those treatments. How does this compare with what we've seen in the past? Work that Perry and others have done suggests that a range they've seen is 0.4 to 2.8. So this is definitely a dry year, but we did get some biomass. It's not out of the range of what we've seen before. So how does this shake out on the graphs? Stick with me here because there's a lot going on. But across the bottom, the x-axis, you'll see all of our treatments, and they have abbreviations so that it wasn't text overkill up here. But it starts with our P in full treatment, and then our 4 functional groups, and then our minus treatments. Up at the top, you can see the species that we have there. And so this is showing you how biomass was by species. And statistically, we use Fisher's protected least significant difference to run these stats. And right here, I'm just showing you how these all compare to the full mix. So the nitrogen fixers, taproots, and minus nitrogen fixers all had less biomass than the full mix, and the fibrous root had more. At the Conrad site, again, we were only able to salvage 5 of our treatments, but the taproot and the brassica both had less biomass than the other 3. I should also point out here that my y-axis is different. Up on top, it's going to 1.5. Down at the bottom at Conrad, it's going to 1. So we learned a lot in 2012. We learned that weed control is essential in cover crop mixtures, because if you're mixing a bunch of species together and you suddenly get a weed problem, you have very little herbicide that you can use to control it because you've got everything in there. We also learned that common vetch is difficult to terminate. We went out a month later, and there it was, just growing along. So maybe we should patent it. We learned that camelina and Italian ryegrass are not competitive in the mix. So then we changed our strategy. In 2013, we simplified our protocol. We moved it to May because we figured we wanted to get better herbicide control prior to planting, and also we realized that farmers would probably be done with their spring planting at that point and be more likely to actually get into the field and plant a cover crop. We set all treatments to an equal seeding rate of 120 plants per meter squared, and we put all seeds together and we seeded them shallow. And then we moved to Thailand. Just kidding. So 2012 was a record dry year. 2013 was a record wet year, and we just saw green everywhere. This is the Bozeman site as it was growing. We got 3.7 megagrams per hectare there, which blows way past the 2.8 high that we've seen before. That's Jeff Holmes out there in the field, and you can see that the biomass is definitely past his waist. And when we would go in to harvest, it was like a jungle. It was his own little ecosystem in there. At Dutton, we got 2.7 megagrams per hectare, which is definitely right up at the high range of what we've seen before, but it's definitely within the range. And how this shook out, again, this is the species at the top, and these are our treatments across the bottom, and then our biomass production along the left. And again, these scales have changed from 2013 to 2012. And comparing everything to the full mix, only the nitrogen fixers and the taproots were less. All the other treatments were statistically the same in biomass production. And at Dutton, we had no statistical differences between biomass production at the site. So what this is telling us is that once we kind of equalize that seeding rate, we really saw very few differences in biomass production. And there's kind of this idea out there that, well, if you grow more species together, you're going to get more biomass. And I'm not sure that that's what we saw here. So that's biomass quantity. Let's talk about biomass quality. We measured cover crop carbon to nitrogen ratios. We separated all the plants in the field. We dried and analyzed them with LECO, and we hypothesized that the cover crop C to N of the P would be less than our full mix. And what happened? In Amsterdam, we did see a small difference. P was less than the full mix, but at Conrad, P and the minus fibrous roots were statistically similar. And at Bozeman and Dutton as well, they were statistically similar. Why is this important? Because we know that the C to N ratio of the inputs will affect whether it's a bacterial-based decomposition or a fungal-based decomposition. Higher C to N in general encourages greater fungal biomass, and that can help to decrease nitrate leaching. We looked at soil water. We took these measurements at the time of cover crop termination. We took 2 cores per plot to a depth of 0.9 meters, and we hypothesized that soil water in fallow would be greater than all other treatments. At Amsterdam, we were surprised. These are the depths. We segmented the results by depths to 0.3, 0.6, and 0.9. At Amsterdam, we were surprised we found no treatment differences from fallow, and that was a shocker. But our thought is that, well, it was just so dry, there was no soil moisture to retain anyway. The story at Conrad was a little bit different. I should mention that Amsterdam is a sandier soil and Conrad is clayier. And Conrad, as expected, all treatments compared with fallow had less soil water. In 2013, again, these are the abbreviated treatments across the bottom. In 2013, all treatments had less soil water than fallow, except for the fibrous root, which grew about 3 metric tons of biomass at that site. So we're scratching our heads as to why there was still the same amount of water in that soil. And then at Dutton, there was a couple surprises as well. At Dutton, we had some unusual results in the lower 0.6 to 0.9 profile of the soil, so we did not report those results. But this is the top 2, 0.3 increments here. And it looks like our nitrogen fixers and our fibrous rooters and our minus fibrous rooters all conserve the same amount of water as fallow. We looked at soil nitrate. Here we just looked at 3 treatments. We looked at cover crop termination. We took 2 cores per plot to 0.9 meters, and we expected there would be more soil nitrate in the fallow than in all the other treatments. Even though peas do fix nitrogen, we know that they first take it up before they start to fix it. At Amsterdam, we got some strange results when we measured at cover crop termination, so we went back and measured in the spring. And again, fallow and pea were bigger than the minus nitrogen fixers and the fibrous rooters. And Conrad, in June, there was more nitrogen in the fallow than in the pea or the minus fibrous root mix. At Bozeman, again, I'd like to point out that our y-axis has changed here. The Bozeman site was just really high in nitrate. This was the one site where we did see some difference between our pea and our full mix, because P had more soil nitrogen, than the full mix. Because there was no difference in the plant tissue, we hypothesized that was because the P had some greater biological nitrogen fixation happening. And at the Dutton site, fallow was greater than P for the full 8-species mix. So how did this work out as far as the spring wheat yields go? In Amsterdam, I would love to give you those results, but unfortunately it was hailed out August 1st, and we would have had to have a Hoover vacuum to give those results. It's kind of a sad day to go out there. But Conrad, we did get a nice wheat crop. And these are the results from the Conrad wheat yield. Again, across the bottom, fallow P, nitrogen fixers, taproots, brassicas, and minus fibrous roots. And we see that fallow had the greatest yield. There was some statistical differences in there, but we don't really want to hang our hats on that data because it was data from one site year. So it's really, I don't want to go into interpreting that too much. When we bring our water chart back up, we can see that water definitely did have an influence on that following year's wheat crop. And if you compared the P yield to the fallow, the loss in yield there was about 9 bushels an acre, which by last week's prices works out to a loss of $59 an acre. That's pretty substantial. All right. So that's the agronomic objective. I'd like to switch gears a little bit here and talk about our biological objectives. And these are really the objectives that I was personally interested in finding out. So it does make sense that an intercrop could yield more biomass than a single species crop. I've got a picture up here of an intercrop. I think it's supposed to be corn and soybeans. Those crops have different leaf architectures, and it could be that they capture solar radiation differently. And also, if there's different root architectures, those could be accessing different parts of the soil profile, which could be bringing up nutrients from different areas. And we know that there's 4 ways, essentially, that plant diversity can affect the soil biota. Biomass quantity. If you're adding more carbon and nitrogen into the soil, that's going to change what's happening. Biomass quality. Is it higher in carbon or is it higher in nitrogen? That will change what's going on in the soil biome. Root exudates and mutualistic relationships. So as we were trying to figure out the actual response variables that we wanted to look at, we wanted to keep in mind that, yes, we could go in and, quantify specific bacteria in the soil, but what is that really going to tell us? We wanted to focus on soil processes. Whoa. So for this objective, we only looked at 3 treatments. We looked at fallow, P, and full at Amsterdam, and at Conrad we looked at fallow, P, and minus fibrous root, because as you recall, our full treatment got weeded out. So the response variables, the timing that we looked at, we wanted to look at soil temperature, and we took soil temperature during the cover crop growth window. So this is Amsterdam 2012. Then we looked at microbial biomass, soil enzymes, and potentially mineralizable nitrogen. And we looked at those prior to spring wheat seeding. We decided that the time of spring wheat seeding was really crucial for our sampling time. We know that a lot of these parameters change over any given crop year, but what we really want to know is what's happening when you put the seed in the ground. And then we looked at mycorrhizal colonization after wheat heading in the response wheat crop. So let's start with soil temperature. We installed our sensor buttons in the spring of the cover crop year to a depth of 5 centimeters, but we only looked at 7 treatments, our fallow, P, and full, and then all of our plant functional groups. We retrieved those buttons in the fall. Unfortunately, in 2012, we had button failure, and so in 2013, we wrapped those in clear parafilm. Our hypothesis was that fallow soils will be warmer than soils in cover crop. And how did that shake out? This is from our Bozeman site, and you can see here that about the end of June, there started to be a noticeable difference between fallow, which is yellow, P, which is in green, and full, which is red. Although you don't see much difference between the pea and the full, you sure see a lot of difference between the fallow and the cover crops. These were the 5-day maximum temperatures. Each interval represents that. And here's where our termination was on the 5th of July. And you can see that we had a good 5-degree centigrade cooling effect for at least a month after termination. And what's the benefit of that? Well, we know that organic matter decomposes faster when the soil is warmer. So if the soil is cooler, hopefully we're retaining more organic matter, and that could just have a lot of benefits as a result of that. Our next factor that we looked at was microbial respiration rate. This was taken in the time of spring wheat seeding in 2013, 3 treatments at 2 sites, and we used a substrate-induced respiration method. And our hypothesis was that the cover crops, let's just turn that around. Fallow should be less than cover crops is the hypothesis. Why is this important? Respiration is a good way to measure just the total microbial population. We know that microbes are responsible for the breakdown of organic matter in the soil, and that therefore they're responsible for soil fertility. But these can be highly variable over the growing season. We didn't find any statistical differences between our 2 cover crop treatments, so we combined them. And we did see a small difference at Amsterdam where the cover crops were greater, but the p-value there was 0.09. At Conrad, there was no difference between fallow and cover crops at the time of wheat seeding. There could have been differences right after the cover crops were terminated, but when it came around next year, we didn't see anything. Our next factor that we looked at was soil enzymes. So why are soil enzymes important to measure? Well, microbes excrete extracellular enzymes, and those enzymes break down organic matter, and that allows for the release of inorganic nutrients, such as carbon, nitrogen, sulfur, and phosphorus. Once that process has happened, those enzymes complex with clay and humus particles, and so they stay in the soil, and they kind of are like a fingerprint in the soil. They're much more reliable measurement than microbial biomass, and also they're very sensitive indicators to early management changes. Whoops. So the 6 enzymes that we looked at some for carbon, nitrogen, sulfur, and phosphorus cycling. Again, these cores were taken at the time of spring wheat seeding to a depth of 10 centimeters, and we did a lot of work in the lab. Ultimately, the results are measured with a spectrophotometer. And our hypothesis was that the enzyme activity in the fallow would be less than enzyme activity in the cover crops. But our results, there's no graphs to show you. There was no difference among all 6 enzyme activities at 2 sites. So that was interesting. We were expecting some differences, but maybe next year we'll see some with more biomass. Potentially mineralizable nitrogen, we felt that was important because that measures the ability of the soil to convert organic nitrogen to inorganic nitrogen. Again, this was taken at the spring wheat seeding of 2013 to 10 centimeters. It was a 14-day anaerobic incubation, and we hypothesized that fallow would have less PMN than cover crops. At the Amsterdam site, both P and full were statistically the same, but they did have more PMN than fallow. And at Conrad, the P was higher, and the minus fibrous root and the fallow were statistically the same. I should mention that PMN is kind of a measurement of how much biomass nitrogen did you put into the soil. So I don't know why the MFR was different than P here. There was the same biomass N addition and the same C to N ratio. One of the final things we looked at was mycorrhizal colonization, and this was taken at the time of wheat heading in that year 2. We went to the field and we collected 4 plants from each plot. We cleared and stained the roots, and then we put those root sections on slides, and then we looked at them under the microscope And wherever the microscope intersection would cross a mycorrhizae, that was counted as, yes, but if it didn't cross one, it was counted as no. So that's how we measured that. Our hypothesis was that fallow would have less colonization than cover crops. We saw no difference, again, between our full and P treatments, so we combined them statistically. At Amsterdam, surprisingly, we had high AMF colonization going on already. We think that might be because it's a very low phosphorus site. It had about 13 parts per million. At Conrad, they only had 11% colonization in their after fallow, the wheat roots did. But after cover crops, that was more like 22. So we did see an increase in mycorrhizal colonization from fallow to cover crops. A good question here to ask, though, is the practical significance. Is that good, bad, ugly, or neutral? We don't know. There's many papers out there that suggest that increased colonization actually increases yields in the following crop. There's some papers coming out of Australia now that say you get increased yields when you decrease mycorrhizae. So there's a big question is how do mycorrhizae function in our soils? We do know that they're very important for soil aggregation, but as far as how they influence wheat yield, there's a big question mark there in our soils. So this is a summary of the short-term results, in case you're wondering. Any place that is blank on this chart means nothing really to write home about, or no big differences between the single species and the mixed species. At one site here, we did see that the mixed species had more biomass quality than the P. We had some interesting soil water differences here at Bozeman and Dutton, where the MFR, the minus fibrous root, had more soil water than the P did. So that's interesting to look at. We did have one site here with differences between the full and the P as far as soil nitrogen goes. One site where microbial respiration differed, and one site of PMN difference, and one site of AMF difference. And then at both Bozeman and Dutton, our fallow soils were cooler than under cover crops. Well, so what? This is my favorite slide. What does this all mean? There's a lot of numbers there, and 2 years of my life wrapped up into that. So what? Are there other considerations that we should have thought of? Other things going on that might be interesting to look at? Well, the first thing was wheat stem softfly control. We didn't really even think of this or put this in our design, but what do you know? Oat is a trap crop for wheat stem softfly. The females lay their eggs in oat, and something about that oat plant either kills the eggs or they can't hatch. We know that we have a huge wheat stem softfly problem in northern Montana. This might be something to investigate. My understanding, though, is it only really works in that April planting window. Another thing that we could look into is pollinators. With the flowering radish out there in the field, I have never seen so many pollinators in a wheat field in my life. That was pretty cool to see. You can see here in the middle of this picture, there's a big bumblebee. And David Weaver came out and he said he also saw the parasitoid wasps that are parasitizing wheat stem softfly. So there may be more going on here than we had originally looked at. Wildlife habitat, we hear from people that antelope like to live in these cover crops. Could we potentially graze these? I don't know. It's early in the season. There might be some issues with nitrates, and even if we grazed, we might have to spray out because it probably wouldn't kill the plants, but could there be a forage benefit there? What's the difference in nitrogen fixation between these single species stands and the mixed stands? And what are the long-term effects? We know in single species crops it takes at least 6 to 8 years before we start seeing really great soil nitrogen benefits. So what would happen here in the long term? Those are my thoughts. If you have any, if you want to know more or learn about more, we do have a website that I've listed here, the Cover Crops website under the Soil Fertility Group. And we have a 7-minute YouTube video that you can watch there that we put together. You could also go to NRCS. They put out a lot of things on cover crop cocktails. I'm sorry I don't have a cocktail for all of you here today in the afternoon seminar. But NRCS has some good information as well. But just be mindful of where that information is coming from. Is it coming from a subhumid or humid agricultural system? Be careful about thinking if it can work in your semi-arid system. And then I'd like to thank all the many people who contributed to this project. There's no way that I could have done this all on my own. Western SARE was our funding source, without which none of this would have been possible. MSU has been fantastic. I'd just like to put a special shout out to my committee, Dr. Miller, Dr. Jones, and Dr. Zabinski, who have been incredibly supportive and patient as I've learned the scientific process. And everyone I've encountered here at MSU has been so helpful and very kind in the whole process. So thank you so much for coming. I really appreciate being here.
I was also remiss in not saying something about the committee structure here for Susan's project. I think more than any other master's project or graduate student project I've been on, this was truly a committee effort. So I'm the one standing up here, but Clayne and Kathy really made this happen. So it was really a group effort. Susan's got time for questions here. It looks like 10 or 15 minutes, and then we'll look after her from there.
I was excited by your other considerations section, right? And I'm curious, in the spirit of a below ground consideration, I thought you'd mentioned that the Conrad showed the most water, right? But you also mentioned it was clay. And then have you thought about just reframing how you present the water in terms of maybe water-filled pore space, right? So if Conrad's a clayier soil, it has a lot more pores. So even though it looked like there was more water, maybe the fraction of the total pores would have been less. Does that make sense?
It doesn't. Let me go back to that slide.
Well, and you highlighted in your main take-home that was one of the big differences that you saw, right? The soil water.
Right. Bear with me as I figure out this roly thing.
Well, just if you have a sandy soil versus a clayey soil, and you're presenting your millimeters of water.
While she's finding the slide, Tony, how do you measure that?
Well, there are easy ways to estimate the porosity based on your texture. Just since you had such a big difference, I think you mentioned Amsterdam was sandy and the Conrad was clayey.
We're getting close. Okay, there we go.
Oh, that's the slide, right?
Yeah.
But if you have twice as much porosity in the Conrad, then that would sort of knock that down a little bit. Does that make sense?
Can you tell me the exact question there?
Oh, so if Amsterdam is sandy soil,
yep,
and you have 170 millimeters of water in it,
okay,
maybe that's a lot of water for that soil compared to 250, say, for your fallow on the clayey soil. But you've got 3 times, 5 times as much pores.
So try and normalize it for each site. Is that what you're kind of saying? Yeah.
Since that's what the roots are seeing. Does that make sense?
Mm-hmm. Good question.
Susan, you mentioned at the end that, sometimes these changes take a lot of time, or at least 5 years or so before you might see changes in the soil, microbial communities, etc. So what are the plans for these plots in the future? I mean, I realize you're finishing up, but just be curious to know where this is going in the future.
That's probably a Perry question.
Yeah, so this is conceived as a 4-year project, and Susan's really engaged in the first 2 years of this cycle. And then if we can scrounge up funding, we'll probably try to find a way to keep the Gallup Valley sites going. It's way too expensive to run run up north on a regular basis. But I think we'll try to keep the two local ones going long enough to make sure that we really investigated this.
Darren.
I'd be curious on your plots where you said you had them weeded out, and so you only reported the ones where you had your plots, because I guess there's arguments out there that weeds are cover too.
Mm-hmm.
And so we're spraying out our weeds. So I would be interested to see your results on your weeds versus your cover crop versus your water use. And I mean, who's to say that weeds can't act as a cover crop? As long as you get them out before generating more seeds to add to your seed bank.
Right, but for downy brome, wouldn't that have to be like?
I'm just arguing that somebody could say that weeds
Yeah.
are a cover crop, and it'd be interesting to see the data.
And you could say,
So it turns out that, do you remember what happened with that, the down-to-grown plots?
well,
weed
it was
data?
like 80% weeds. I remember that.
No, but relative to the wheat yield. And we did measure them.
You did. Good.
Yeah.
Oh yeah.
And the yields were quite high, actually.
This is sort of what's happening in the soil. That's right.
There's a bunch of stuff we didn't show you. That's a good question. I mean, and it might just influence like mycorrhizae. I mean, if you have wheat, a weed there, that's probably going to be. It might be a mycorrhizal plant. So there you get a benefit.
I say get rid of them. I'm just playing devil's advocate.
That's a good question. And when you start dealing with all these plants and the complexity you start to realize how many questions there are.
What happened there, Darren, is this site was supposed to be sprayed out for us by the cooperating farmer, and we show up to seed, and it's been sprayed yet too. Not only that, he's not taking delivery of the sprayer until tomorrow. So we seeded, and then he tried to spray out after we seeded, and it just didn't quite work.
I don't know if you saw that, or maybe I didn't see this before, but I know that a lot of times brassicas have been suggested as cover crops for their allelopathic action. Did that at all feed back onto some of your wheat yields, or is that something you're going to see in this next phase?
We don't know. We know that brassicas are non-mycorrhizal hosts. And in our wheat yield slide, I had something going on that I couldn't explain, is that the brassicas. Yeah, brassicas did use, some water, but out of everything, they really didn't use as much. And there was only like 0.2 megagrams of biomass that they produce. And these two produce a similar amount of biomass, cover crop biomass. So why does this one have less wheat yield? I don't know. Harry said, don't mention that.
So what are we talking about?
Well, it's just that it's one-side-year data, so, but it was interesting, and I think we'll be looking for something like that next time.
Well, Susan, congratulations for the presentation. My question is related with Jane's question that if you knew in your introduction that a single cover crop, you cannot see the effect after 6, 7, or 8 years in the next crop, cash crop, for example, and you were expecting that with the cocktail you could see something in just 1 year?
Yeah, then why did we even start looking at it right now, kind of?
Well, my question is why we have to wait so long to see improvement in the. So in your introduction, you said that previous author has seen that you cannot see an improvement in your cash crop the next year after a single cover crop. It has to wait 6 years.
Okay. Right, so why?
Why?
Basically for the nitrogen benefit. So, the cover crop grows and then it dies, and then it just takes time for enough cover crops to grow and die, so to get enough biomass on the soil, and then those leaves, that leaf tissue, slowly breaks down, and it breaks down slowly in our climate because we're cold and dry, basically. And so once it starts to break down, that breakdown from the organic matter provides an inorganic nitrogen benefit to the soil. But it takes time.
Sort of feeding off of this, and maybe more broadly than just you, Susan, did you have expectations at the beginning with a mixture to have that effect speed up because you're feeding the soil a more diverse food source? Did you have any expectation for what that kind of delayed effect would be?
I think I was definitely over enthusiastic at the beginning of the project, but that's typical for me at the beginning of any project. So, so yeah, I was hoping to see, especially in the enzymes, I want, I was hoping to see differences there that might give us a clue as to what's happening. But it seems that they're, functioning very, very similar to the single species.
Seems like one of the potential benefits of a diverse mixture would be stability or resilience.
Mm-hmm.
Did you look at all at sort of the variability within your treatments and if that differed between your mixes? So, as a concrete example, P might do awful in some sites, some years. But
Right.
if you had a mixture, you have sort of an insurance policy that something's going to do well.
Well, that's why we designed the study with 2 plant species per functional group, just for redundancy. But we didn't look at that factor specifically, but it's a great idea. Is that, we all know all the pests and diseases that could come our way. So what if P weevil gets your crop? Well, at least you've got something covering the soil that'll cool it.
We talked about in Perry's class how sometimes it's hard to get an even cover of the cover crops with the cocktail because the seed size is different. So some will settle to the bottom of your seeder, and you'll have patches of one thing and patches of another. Did you have that issue?
We didn't, just because we have a really tiny seeder that's about 6 feet wide. So we didn't use if you remember the photo in there, the no-till fellow, I can't remember how wide Carl's seeder is. 40 to 50 feet.
Yeah.
So with this huge tank on the back. So sometimes they'll stop the tractor, get out, and mix it. But we didn't have that problem. So, but we're just using much shorter plots and much smaller equipment.
I think this has been great, and I was really intrigued by your enzyme results. And I'm curious if you used spikes and then looked at spike recoveries to figure out whether you were really pulling all of the enzymes that you were trying to quantify.
I'm not familiar with spike and spike.
Well, so you would add that enzyme in and then see if you could get out that amount of enzyme. Does that make sense? Like, how do you know that you got all the enzymes out with your extraction?
I don't know. I hadn't thought of that.
Quit asking questions, Tom. Sorry.
Because I talked with an expert who said she thought it sounded good. That's the truth. So.
One more burning question.
Can you summarize what your best argument for a diverse mix is?
My best argument for a diverse mix? Zach.
Or would you like us to save that one?
My best argument for...
Or is there an argument?
I think the best argument for using a diverse mix will be the grazing potential and how you could integrate livestock into a cropping system. So you'd have some benefits in your soil, because it'd have some cover on it. You'd have a forage benefit to the livestock. You'd be saving whatever forage that your livestock have been grazing over in a pasture. They can now graze in here. There's some manure benefit on your land. Everyone that I've talked to about this, who's using this technique, say they really see benefits to the bottom line when they integrate it with grazing. So I would say that is the biggest pro for these, and I would say that the biggest question then that I have is how. Oat is great for sweet stem socklight trap, but it's also known as